Okay, so this next little video here is say podcast. This video here is going to talk a little bit about the history of genetics. Okay, it's good to know, kind of see where these things are coming from. It kind of starts with this guy called Gregor Mendel. He was a priest, typically back in the Middle Ages. Anybody that did science, they were typically Catholic priests. This guy was, he worked in an abbey, and he was a gardener. He was on the staff there at the abbey, and he did some gardening. And with any gardener, you try to make better and better plants. And he was working with what's called a pea plant. Kind of a pretty little flower. You may actually grow these in your gardens. A little sweet pea plant, have a nice purplish flower. And he was noticing that some of the pea plant pods. So a pod is a little capsule. It's got some peas in it. Okay. He was noticing that some of these peas were green. Some of them were yellow. Some of them were wrinkled. And so he was kind of wondering why some of these plants were starting to have different pea plants. And so he noticed this. He made an observation. Sorry, that green's not really showing up. These are two different colored greens here. Maybe that's better. Maybe that's worse. I'm not sure. Try this one again. So you got some wrinkly. And so what he was taking the parent plant, and they would crossbreed them. So we're talking about sexual reproduction. We had the male parts of one plant and the female parts of another plant, mixed them together, and you would get offspring or these seeds. And the seeds might have been green round. They might have been kind of a yellowy round. They might have been green and wrinkly, and they might have been maybe yellow and wrinkly. And depending on which plants he crossed, he got this kind of combination of things. So this guy, Gregor Mendel, was like, well, what the heck's going on, okay? He ends up developing this idea into what's called a Punnett Square, okay? And so this idea of a Punnett Square is I have different percentages of offspring. And he started to see these percents. He said, like, well, yeah, I've got, 25% of these, and I've got 25% of these, and I've got 25% of these, and 25% of these. And so why do I have that, like, perfect ratio that started to appear? Okay, it wasn't really 25%, but for simplicity's sake, it was a really nice ratio, and it was the same ratio every single time. And so he's wondering why. Why does this happen? Okay, and so he develops this idea of a Punnett square. Okay, Punnett square. Okay, the Punnett square is a tool that we use to calculate the chances of having a certain trait appear. Okay, now we work with the genotypes. Okay, so let's kind of set this back up. Just review some of the terms. The trait that, let's say, we'll deal with, and I'll write this maybe over here so you can see it. The trait, okay, let's deal with eye color. I like eye color. It's pretty easy to work with. Now remember, sorry, is that glare in there? Sorry. Eye color, it's right in the glare. What you have here is, we're kind of simplifying this stuff. This isn't real life. It's close to real life, but it's not quite real life, okay? The phenotypes that we can get blue, and we can get brown eye color. And we're going to say that blue is dominant, and I'm going to say brown is recessive. Okay, so we talked about dominant and recessive. So if I know blue is dominant, I have two options here for the genotype. Okay, so if it's blue and it's dominant, I have one option. It could be homozygous dominant. Homozygous means it's the same big. It could also be heterozygous. I'm sorry, I'm losing my mind here. The alleles we'll use. Let's use B. Okay, B is a pretty simple allele. So we have homozygous dominant and we have heterozygous, okay, for the dominant version. So these are going to be my blue ones, okay, because with dominant, I only need at least one. So that has at least one. It's got two. And this one has at least one. It has one, okay. The recessive trait is going to be that lowercase, and the only way I can get the recessive is if I have two lower cases together. Okay, so just kind of a quick review of the terms. The allele I chose is B. It could have been any allele you want. You could have used E, okay, capital E or lowercase e. Anything would have worked, maybe E for eye color. But anyways, the trait we're working with here is eye color. We have a phenotype of blue or brown. I said blue was a dominant phenotype, brown was a recessive phenotype. And that's important because it helps me figure out my genotypes, because dominant could be capital or it could be capital lowercase. We called that homozygous dominant. We called that heterozygous. Brown was recessive. The only possible thing I could get was homozygous recessive, which is lowercase. Okay, so you got to kind of clear that up before you work with these Punnett squares. Okay, Punnett squares. A Punnett square is a tool that helps us figure out what's going on. Okay, so I'm going to use two different colors here to kind of sort this out. Each box could be an offspring. Okay, so every time there is a sexual mating, so mom or dad, so we'll put mom up here, we'll put dad down here. It does not matter. It could be dad and mom, just male, female. Okay, this is sexual reproduction. Mom donates one allele. Okay, so let's say mom starts out, she's got blue eyes. Okay, she is heterozygous for blue eye color. Okay, let's say dad is homozygous recessive. He's got brown eye color. So homozygous recessive, he's got brown eye color. Now, because, and we kind of glossed over this with meiosis. Do you remember when I split those papers into 4 pieces where we had 4 different gametes, okay? In sexual reproduction, we have to make gametes. So mom has all of her genetic information. How much is she going to donate to the pot when we make another offspring? She's going to donate 50%, so half. So half of this is a big B, or it could be a little b. So we like to write that like this. So we're going to split those up. Okay. Now I'm going to change colors here for dad, make it easier to track for you. Dad's got the same thing going on. Because of sexual reproduction, we have to make a gamete. A gamete is half of the DNA because dad has little b, that's all of the DNA. Okay, half is going to go into one gamete, and half is going to go into another gamete. And so my options are B and B. So little b, he only has one option. Okay, so they're going to split. Okay, this principle is called the separation of alleles. Okay, it's very important in genetics because in sexual reproduction, half of the material is going on, and that's what we're signifying. So half of mom's is going to go in here, and half of mom is going to go in here. Same thing, half a dad, okay? And so we kind of play the little Connect 4 game. Maybe you're not familiar with Connect 4. I suggest playing it. It's a fun game. This letter is going to go down all the way, okay? This letter, same thing, is going to go down all the way. So it's going to fill in each box, okay? Now dads are going to sort same way, so they're going to go here and go here and here and here. So nothing terribly complicated. You'll get much better at it, okay? General rule of thumb, we do not write little b, big B. That is not proper, okay? We always write the capital first, okay? And the reason for that is when you start combining alleles, it's much easier to see when different combinations of genes come together. So I have 1, 2, 3, 4 genes listed there, and they always start with the capital, okay? So that's just kind of a proper thing here, one that you should take note of. We do write capital letters first, okay? So now after I've kind of made this all confusing with these lines here, get rid of these lines. I end up with 4 different possibilities. And so if you look at mom and dad, they definitely have a kid. That's a 100% chance of having a kid. Well, it's 25% chance of having this kid. It's 25% chance of having this kid. It's 25% chance of having this kid. 25% chance of having this kid. Okay? And so I might ask you the question, what is the percentage of mom and dad having a blue-eyed kid? Okay? And so, oh, blue-eyed. What's blue-eyed? Well, blue-eyed could be big B, or it could be big B, little b, because it's dominant. Okay? So what's the percentage? Well, do I have any big B's? No. Well, how many big B, little b's do I have? Right here. So I have these two are going to be blue-eyed, 25% chance plus 25% chance is 50% chance of blue eye. Okay, and what about brown-eyed? Same thing. You look back over here. Brown-eyed is only one option, but turns out big or little b, little b, little b, little b. These over here, okay, these are going to be same deal. 50% brown-eye. Okay, that's generally how it works. All right, now I'm going to do another one for you. So maybe take a picture of that, stop it, rewind it if you want to hear that again. I was trying to be very careful with the terms I use, and we'll go through it one more time. Okay. Let's say I use purple. Purple seems to show up a little bit better. Let's say I want to look at the trait of tall. Okay. So my trait here is tall. I'm going to use the allele T. Okay. I'm going to use the phenotypes. I'm sorry, confusing myself. The trait here is height. I apologize. Height. My phenotype could be tall, or it could be short. And I'm going to say tall is dominant. I'm going to say short is recessive. And so my genotype for tall, because tall is dominant, it could be big T, which is homozygous dominant, or it could be big T, little t, which is heterozygous, or it could be short, which is recessive, meaning it has to have little t. Now, a little side note here, try to choose letters that's easy for you to tell the difference between capital and lowercase. S is horrible, because a lot of your capital S's and your lowercase s's look the same. So you either need to be extremely careful about your capital and lowercase, or, interesting little trick, you can put the lowercase in cursive sometimes. That makes sense. So I have a dominant S and a recessive S. You don't want to make your T's nice and sloppy like that, because now which one's dominant and which one's recessive? Well, this one kind of looks like a lowercase t. This one kind of looks like an uppercase t. And that's why you'll see me do these little kind of cursive T's for lowercase and a very clear, crisp capital letter for my capital letters. P's are also hard, okay? So instead of doing something like that for dominant recessive, I might do capital P and then a cursive P to show dominant recessive. So be careful with your genotypes, letters you choose. Again, it could be any letter, unless I've specifically given it to you on a worksheet, okay? So the question is, maybe work this out for yourself. If a homozygous recessive male bred with a heterozygous female, okay, what is the percent of having offspring tall or short? I mean, you could put this either way. Okay, so that's our question here. If a homozygous recessive, so what's that? Homozygous recessive. Homozygous recessive is little t, So does that mean is that a tall person or a short person? Short person, good. Okay. So homozygous recessive male breeds with a heterozygous female. What's heterozygous mean? Well, there's only one option here. Is heterozygous tall in this case? Yeah, it is. So we have a heterozygous female. And then we want to know what's the percentage of having a tall offspring. So let's do the Punnett square here. So I'm going to start with my box. And my homozygous recessive is a male. It doesn't matter where it goes, but I know that it was TT, little t, little t. Okay. And then the heterozygous female was capital T, lowercase t. And I'm going to drop down these. So I have lowercase t, lowercase t, lowercase t, lowercase t. So I just drop them down. And then I'm going to have these go across the sides. Capital T, lowercase t, lowercase t. Okay? And the question here was, what's the percentage of having tall offspring? So what is tall? Well, tall could be big T, or big T, little t. And so I count these up. So I have 1, 2. So these are possibly tall. So that could be 50%. And then I would ask the other question, so what's the percentage of having a short offspring? And you would see that at the bottom here, little t, that was short, that was 50%. Okay? So that was Punnett squares. You're going to be doing some labs going through all this stuff pretty much all next week. You'll be doing examples of these, and we're going to start slow. We're going to start with this one here. This one talks about having your traits, your alleles, and you've got to figure out the possible genotypes, and you have to figure out possible phenotypes. So it's very important you know those terms. Okay. And then the second procedure, what you're going to be doing is you're going to be answering what's the percentage of having only one eye, and there's kind of these monster type things. So you're going to use a table, and you're going to use this Punnett square method I just showed you to figure out some percents. So you're going to work with the terms, you're going to work with Punnett squares, very basic. Okay. Next assignment is going to be the pink one here. This one, it kind of goes through again how to do a Punnett square. It gives you a step-by-step procedure, and some of these already draw their boxes, and you're going to have to answer the questions again. Homozygous dominant, heterozygous, homozygous recessive, the characteristics, so the phenotypes here, the genotypes, the traits. You're going to have to understand all that stuff. Okay. The questions here. How is it possible for two parents of brown eyes to have a child with blue eyes? Oh, man, that's a good question. Well, could you have two heterozygous? What would that look like? What would two heterozygous? Maybe that was brown, or blue. Could you have brown? Well, if you quick do a Punnett square, is it possible? It is possible. It's not very common, but it's possible. So all you out there asking, well, why am I the only one with red hair in my family? Well, that might be the situation. Okay, so we're going to go through that as some practice. And then there's some more practices. They get a little bit more in depth. So we've got a yellow and a green sheet here. Same thing, read through the directions. Mrs. Hardy can help you out. If you have questions, you need to be watching these videos. Okay. Eventually, towards the end of the week, possibly even next week, you're going to get into a more complicated lab where you're actually going to make a monster. Okay, so these monsters, you can play with this stuff. Do you have spots or mouth size or number of legs, number of eyes? There's some different things you can do with monsters, and then you can even breed your monsters with other monsters in a classroom, and you can come up with new offspring. So you're going to kind of be playing with that stuff for the next little bit. If you have any questions, please email me, but I would suggest watching these videos again. Finding other YouTube videos would be a good idea also. The key is the terms. You need to know the difference between, where do they go? Traits, alleles, phenotypes, genotypes. The Punnett square is this tool. You need to know how to work through this tool. You need to know the percentages of each of these boxes. And this right here, the phenotype, genotype, going back and forth, is going to be probably the most difficult part of it all. Okay, and it all comes down to that first part where we were talking about how in sexual reproduction, half of mom's DNA is an option, or half of dad's DNA is an option. So you have half of mom and half of dad, and they come together and you get a whole. Okay, we talked about fertilization and called a zygote. That's why this works, because I'm showing half of mom and half of dad going through these Punnett squares, and when you make those combinations, you get some options. Okay, so there you go. If you have questions, shoot me an email.